recording and we'll carry this off.
Mhm.
Well,
Mhm.
welcome everybody back to another webinar we have today. It's fantastic having all of us joining with we have Compost Academy and Sustainable Zion
Mhm.
Masterclass crew. We have Keisha Ernst, joined by Brian Vag and I, and today we have a very special guest, Juan de la Serna. Juan is a soil professional, soil consultant, and composter based in Europe. And I was really excited to bring on this webinar because he spent years kind of creating these case studies and actual having on-farm trials about using compost and biology to turn farms around. And he's seen some very impressive results. And so I think it was great to show these results to everyone because a lot of people in the soul space they're doing the work, but they're doing it silently, and it's really time to show their work because it's really mind-blowing the amount of dedication it takes to actually turn farms around using soil biology. And so Juan's going to share with us today about how far you can go working with soil and working with compost and the results that you can have in the real world on the ground on farms with soil biology. So, Juan, I'm going to let you take it away here, and it'll be awesome to share your story with the world.
What an introduction, really. Thank you so much. Let me share my screen real quick. Let's go.
All right.
Alrighty.
And I'm going to pin you too, just so you're the main speaker here.
All right. Do you see my screen?
Uh,
Yeah.
Yeah.
oh yeah, sorry.
Yep, we can see it. Yeah,
You can see
Yeah.
it. Okay, great.
yeah.
Okay, perfect, guys. All right. So my name is Juan de la Serna, and I studied bioengineering in Belgium and focused on tropical crops as a master's. And I had access to, I would say, the last generation of really, really good professors with 14 years of experience in Ethiopia, in farming systems in Africa, smallholder farmers, etc. So they totally blew my mind. And I was lucky enough to be able to do my thesis in Mexico about conservation agriculture systems, where I studied the weeds in conservation agriculture systems. So without tillage, with crop rotations, and um, basically seeing how the weeds would change. And we, surprise, surprise, we saw some differences. And after having studied this very, very, yeah, intensive science package, I decided to, well, to take matters in my own hand, take my backpack, and just leave and learn by doing. And so that's what I did. And it ended up being a trip of 3 years in South America. And basically, I've been working with microbush compost for about 5 years, maybe a bit more. And I created this company called Resolutions, which stands for Regenerative Agriculture Solution. And this is me in a profile pit. Here on the right, you see a beautiful glaze soil. Basically, the gray part is where the water table comes up, and that was in cacao field. Basically, I love to dig into soils. People laugh about it, but I don't care. Like any true soil nerd, you gotta go through it and continue. So yeah, for me, it's really important to understand that what we see in the world right now, it's the norm. So it's normal to see this in our TV screens or when you go out, that the soil is completely degraded. When it's dry, well, it's normal, it's dry. Well, yeah, well, maybe there's no cover on the soil. Or when it rains, well, yeah, all the soil is washing away because of the rain. Yeah, it's actually not because of the rain. So I just wanted to tell you, okay, it looks normal. This looks like the norm, but it actually isn't normal. And so, well, the big question is like, how do we create farming systems that provide food security, restore soil fertility, and reduce our impact on the environment? Because, I mean, we are disturbers. We are late climax species. We are supposedly the smartest individuals or species in the world. But in the end, we actually destroy the whole motor that is feeding us. So surely there must be a way to work with nature and to be able to feed ourselves without destroying the motor that feeds us. So I'm gonna go through the aha moments I had, like basically mind blown moments, where I got this Swiss farmer that immigrated in Brazil called Ernst Goetsch, where he mentioned that the natural ecosystems follow the natural succession of species. I mean, that's not his invention or his observation, but that's really something that he coined in the sense of that he built a system, an agroforestry system that mimics this. And I thought it was incredibly interesting. Of course, there was Ernst Goetsch, there was Geoff Lawton. There's so many people on YouTube who I mean, I basically got addicted to watching YouTube videos about soil restorations, and my friends just couldn't hold it anymore. I mean, yeah, I still do. I'm still very addicted to this and watching so much content. But basically, the really interesting point is where two scientists talk about the same thing, but in a different way. And here enters Dr. Ellen Ingham that says the same thing, saying, okay, well, along the natural succession of species, soils are increasingly dominated by fungi. So basically, you have more larger scale or longer cycles plants that join the plant succession a long time. But actually, the soil is also changing and it's going to be more and more fungal. So I thought, like, okay, they're saying the same thing in a different way. So I thought, like, wow, this is mind blowing. I gotta understand this more. And I wanted to add another one that really blew my mind is from Claude Bourguignon, which is a French microbiologist. And he was saying, like, look, guys, if you want to save the tigers, well, save humans first, because who are we to say in the Western world, okay, we gotta stop deforestation, guys? Like, yeah, yeah. Well, the guy that just planted his maize field and its fertility is just not, I mean, the soil is just depleted because they are on tropical soils and they planted it for 3 years. They got no fertility left. Well, they're gonna go in the natural ecosystem and they're gonna fell all the trees because there is a good soil, there's still good soil left. So find a solution for humans so that you don't have to find solution for tigers or whatever, pandas, whatever you like, but also insects, whatever. You get my point. We have to find solutions for humans first, because that's the way we can preserve our beautiful biodiversity. So I thought, okay, well, I'm a nerd. I studied in university. I have no idea what I'm doing, so I'm just gonna learn by doing. And I decided to take my backpack. I actually worked in a bar for 6 to 12 months, earned a bit of money, and took my backpack, and there we go. So I went to Brazil, to Bolivia, actually trying to find teachers, people that could teach me, and yeah, again, learn by doing. I ended up in Guyana working with acai in a lot of communities, but yeah. Anyway, so these are a few slides from Peru. I met a lot of smallholder farmers. I found this amazing guy called Albert Merino. He created this company. He was completely convinced from the start, and he helped those farmers generate more income through soil fertility. And I just added those soil profiles so you get an idea of what we're working with. Like this is deep Amazon stuff. Look at this small crust. I mean, it's what, 2, 3, 5 centimeters, 2 inches for our friends over the pond. And it's just really small. Like they don't have so much fertility. And this increment in fertility, this black layer here that you can see, is really through his management, where he put some cover crops in cacao fields, as you can see on the left picture. This is a plant called kudzu, basically increasing soil fertility. And as you can see also in the color of the soil, it's completely gray below. So that means that the water table is going up. So you get this iron oxides, Fe2, Fe2 plus. I mean, there's a lot of pluses in there. Fe3 plus on the top, Fe2 plus on the gray side. Basically, tough conditions. Darwin here, they have the most amazing names. We got Darwin. I also met a guy called Stalin. I also met a Stalin in some community. Basically, yeah, just yeah, you'll get it if you're there. And this guy was working on a soil with 3.5, a pH of 3.5, like just really on the deeper Amazon, really tough conditions. So of course you put some pineapples. I mean, they resist a lot to acidity, etc. Yeah, so really great people, and they really want to learn. It's so cool. And in my journey, I also met Raul Conde, which is a tattoo artist from Berlin. I met him on the market. I don't know why or how, but stuff happened. And he has some land in Chinao, which is also in the region where I was. And I said, like, look, I have an idea to restore your field, which is the prairie you see. I mean, this degraded field there. And I said, like, look, we're gonna do terraces, we're gonna do some tropic agriculture, let's do it. So that's what we did. And I designed this thing, this design that you see here, where the idea was, okay, like how can you generate income while preserving soil fertility so that you don't have to, I mean, let's show a model for people so they don't have to always take a piece of the rainforest every 3 years. So you start with harvest of maize and beans, so you get already a first income. You harvest papaya and moringa after 8 months. 12 months after, you get banana, curcuma, ginger. And after that, from 3 to 8 years, you get cacao, lime, soursop, ginger, etc., etc. And after 8 to 25 years, you get this beautiful food forest. So please copy this design. It's all out there. So just or change it or call me. So I mean, basically, it was my first trial, and so we did. These are the beans that you can see. So we mowed in between the rows and we put this fresh biomass on the rows on the terraces, and we also make sure that the topsoil was put on top of those rows. So yeah, so that we got good fertility. People told even the farmers told me like you're never gonna harvest stuff if you put some how do you call it like this grasses because rats are going to go in, etc. Nah, it was a beautiful first bean harvest. And if you're interested, I cannot go into detail on this one, but there's a PDF on LinkedIn, so just download it. It's all open source. And in the meantime, I reached out to Elaine Ingham saying, at that time it was called Environmental Celebration. So it's a really, really OG shit, like really in the 2018, really like it's a long time ago. And I said like, hey, dude, I have no money. Can you please give me the contacts of people following those courses so I can ask questions? And they were super nice. They told me, well, you know what? You can just do a project, do something yourself, and we'll give you the courses for free. So I did, and that was my project. And as you can see, we did some compost here. We did some compost extracts, I mean, Peruvian style. Also, we got the farmers involved. They looked at their first nematodes, and by the way, me too, first nematode moment. I think everybody, I mean, nobody forgets that moment. Kind of like, yeah, where were you when you saw your first nematode, right? And below on the right, you see, yeah, I just bought a toolbox, and this is my microscope ready to go on a freaking truck and be in the mud and whatever. Like it's just yeah, going into those communities. It was quite far away actually from the city. And so I made some compost tea, and yeah, the place was really like 30. I don't know. It was a crazy angle. I just was working out like crazy by working there, and we had no way to bring this liquid up there. And also to make the compost tea, we just bought a compressor in Lima and took a how do you call it like a trash bin, modified it, some tutorial on YouTube, and there it was. And we put those buckets on some mule because it was the only way. Yeah, anyway, I just love this picture. And my first realization was, okay, well, I gotta tell this guy that works 12 hours in his coffee plantation. Coffee prices go up and down. Now they're up, but they're really down when I was there. And I had to tell him, like, after 12 hours, like, hey, dude, it's time to turn the pile. Like, it's 55 or 60 degrees. We really got to turn the pile. The guy just had a 12-hour workday. Like, I mean, ese gringo el loco. I'm sure he thought that, didn't say it, but definitely, I mean, come on, you got this white dude just like, anyway. So I realized like, okay, well, we really need to find a solution to this turning thing, or it needs to be easier. And so we went direction EMs and stuff like that, but I really like the idea of compost and aerobic compost. And yeah, just as a side note, this is also Marlena, she's such a lovely lady. She cooked me my banana, my fried anyway. She was such a beautiful lady, and she helped me. Yeah, she made her compost herself. She was really, yeah, super involved. Basically, going back to this idea of it has to be more simple. then I came across to this Johnson Su. And so the Johnson Su method, I was super skeptical. I thought, like, hey, you need to get this high temperatures, etc. It's not going to be okay. You're not going to have those pathogen reduction, etc. And I really actually brushed it aside. I thought, like, yeah, he just doesn't know what he's talking about, and blah 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 blah. I just followed the course of what they told me, right? And I tried it out. I thought like, okay, it's actually quite interesting. Let's just not brush it away. And it looks like he's having good results. And so I tried it out. And also like biodiversity was insane after 12 months. And for me, I mean, just with my look at it now that I'm working with this since a long time, it's an absolute game changer. This is a different thing. It's just insane. Like the microbes you can get by just not turning the compost. I mean, you can just a side note here, you can have really high quality by also turning the compost. I'm not saying the contrary. And you can make much more. So it's really a question of like, okay, I get much more quantity. Much more quantity and just a little bit less quality, or I get this insane quality and smaller quantity. So I'm not saying one is better or the other, just saying, hey, it's a trade-off. So of course I tried it. Well, this is not my first try, but I tried it with an IBC. I tried also with this classical construction. Rats came in, it froze. Yeah, I mean, yeah, it was a mess, but yeah, it was also a nice learning experience. I tried it with an IBC. It worked fine also, except it really froze overnight. I was in Germany at that time, and we got what -10 or something. I just had an ice block of leaves and composting materials. So yeah, the temperature part is definitely a big one. And but the thing is, like Johnson Su really shifted my mind. Of course, Ellen Ingham was talking about inoculants and microbiology, but to have this compost applied in such small amounts made me think like, or at least made me realize that micro-rich compost is actually not a fertilizer, it's more like an inoculant. So of course there are some nutrients, there are nitrates, ammonium, etc. It's all there. But the important part is, okay, where are the workers? Like who's there? There's so many microbes in this type of compost. I think there's more than 500 species of fungi, protozoa, nematodes, many more critters. So it's really about the biology that you can achieve with this compost rather than the nutrients. Of course, the nutrients are definitely important. And so who's there in this compost? Well, you got amoeba, you got nematodes, you got fungal hyphae, you got protozoa all the way, you got everything. Bacteria also super important. And we did a lot of trial and error. And I tried a lot of different recipes and trying to nail it down. And it was such a, yeah, it's just a lot of work, let's say. So I wrote like after trial and error, but it's actually more errors than trials. Oh no, I mean, obviously it's the same, but a lot of errors. And we got this. I sent it to a lab because, of course, like I'm definitely biased. I would think, oh, my compost, blah blah blah, is the best. But I don't think that actually. But I thought like, okay, well, you need to have a different. You have someone else. Need someone else to look at it because you're definitely biased. Like it's the same as if you do a trial. Like by the way, the trials that you're gonna see, I'm not harvesting the data. I'm just look there to see the data is really well harvested. But you gotta have this distance. If you're doing experiments, you have to, because you're biased, you have to be there, but make sure you don't collect the data. It's really important. Anyway, we got 11 times more fungi, 100 times more protozoa. I mean, I think we got 1 million or something. And it's really something not to forget that protozoa are so important. Like we focus on fungi, etc. And I'm guilty here because they're really cool. But protozoa are so important, and we ended up with a fungal to bacterial ratio of 30 times higher than the minimal required. So minimal required is in function of the Soul Food Web benchmark because they actually started the whole thing. Like they just said, like, okay, well, 135 for bacteria or 135 micrograms per gram of compost will be good. If you're gonna go on fungi, of course you can go much higher, that's better. But yeah, so I was really amazed by those results. just a moment. Mm. Of course, don't don't think like, okay, well, wow, this compost has a fungal to bacterial ratio of 10. It really depends on how many bacteria you have. It depends on the state of the compost. It depends on the lab. It depends on so many things. So I'm not saying here, oh, well, this is the truth and amen. I'm just saying like, hey, this is what some guy measured. Actually, it's super Francisco is called. He's really cool. He's doing great work at Orgo, but he measured this. We sent it to different labs. We had more bacteria. So we also had a lower fungal to bacterial ratio. So yeah, just as a side note, but the fungal content was similar. So it was from 1500 to 1800. So we really got constant on the fungal ratio, the fungal biomass. And so how does it look like? Well, it's incredibly fluffy. Like we get this really fluffy material. And during the composting process, of course, it's a joy. You see all those hyphae going crazy and digesting this material. So yeah, I just love that stuff. And in the microscope, it looks like this. So you dilute it 1 to 10 and magnification of 100 for all those microscopers here out there. And we look at this. So we, of course, got those fungal hyphae, but you also get those spores, which, by the way, are not countabilized or they're not counted in some microscopy assessments. So you also have to count the spores, right? And so just so you get an idea, between the compost that we can produce, that is possible to make, and the compost that you would buy in any shop that sells you compost for your beautiful plants in the summer, well, you get a huge difference. Like on the left, you see some fungal hyphae that are quite long, as you can see. It's the same magnification, same dilution. And on the right, you get this bacterial soup where it's just like hanging out there, but you don't have this structure. You don't have this aggregation between microbes and organic matter. You just have like this soup, which is, it just doesn't contain, in my opinion, or what I think it is, it doesn't contain those microbes that can build soil structure. And this is really what you want. Fungi, bacteria, etc. And since I think everybody should see a nematode in his life, well, here it is. So this is a young compost that I analyzed, no, that I produced, I'm sorry. Um, and yeah, it's feeding on those beautiful hyphae. Anyway, so why compost? And I just talked about it just before, is you want those microbes that build soil and soil structure. The soil microbes are going to build soil structure and soil aggregates. They're going to build gluing agents. They're going to make the bricks, so the bacteria make the bricks, like Elaine Ingham told us. And the fungi are going to be the cement between the bricks, and they're going to build the soil structure. So you need all those little critters together. But just so you realize here on the right, you see this fungal hyphae that are really you can see them on the electronic microscope, so it really gives a visual feel to it. And of course, microorganisms are central, if not the most important thing in carbon. So you want to save the planet with CO2. Yeah, if CO2 is something that makes you anxious, well, just make compost, right? So they're going to transform the organic matter. They're going to transform this organic matter into either into CO2 as respiration and create energy, or they're going to transform it in labile carbon, or even better, when they die, mostly fungi, when they die, they have such a complex carbon, how do you call it? Carbon connections or carbon, how do you say this? In chemistry, bonds, yeah, so they have chitin and stuff like that. They're extremely stable, and so they're not degraded, but they're still kept as carbon, and so you get this also bacteria, of course, but you get this necromass that's going to attach itself to minerals of the soil. So if you want to put more carbon in the soil, well, you got to work with microbes for sure. And this is my little rant. I apologize in advance, but like, come on, guys, if we're going to buy a soil inoculant with 10 different species of, I don't know, PSBs or phosphorus solubilizing bacteria, we're going to buy some mycorrhiza. I mean, nothing against mycorrhiza, they're great. But like if you buy this cocktail of 1 to 10 to 20 species, it's not, I mean, it's great. It's better than nothing. But when you look at the diversity of compost, it's just a different game. It's just a different thing. And it's also adapted to your soil. So it's just this microbial inoculant that's so diverse and also redundant. You want redundancy inside of your inoculant. You want different groups of bacteria, fungi, etc., to do the same function. So if one dies, well, the other one takes over. And this is a study. This is like pictures from a study that I really like from a French team, and they analyzed the connections between forests and vineyards and passing through grasslands and crop systems. And you see the connections in green between the different microbial communities. And when you go from forests, which is basically the biome we live in Western Europe, northern Western Europe, and vineyards that are usually quite intensive, you see that the connections are lowering and lowering. So you might get actually a higher diversity in vineyards. So you could count bacterial diversity or fungal diversity, but the connections are not there. So I'm thinking, okay, well, why don't we introduce microbes that have these connections together, that know each other, that communicate with each other since you started the compost, basically. So diversity is great, but connections are more important. And yeah, it's just so interesting. So let's go to the meat of this presentation. It's going to be about my field trials today. We use compost as a seed coating, as compost tea or compost extract, but also as a substrate, as you will see in market gardening. Mm. So it was COVID in 2020, and I bought I bought I think 3 caddies worth of toilet paper so I could like survive the apocalypse. And in the meantime, I rejoined my friend that was a farmer in Belgium, and we tried the seed coating of Johnson Su, and we got really good results. We just tried some seed coating, and by the way, there's a PDF that you can download for free also to see how we did it, and we got really good results. I think it was beginner's luck. And after 64 days, we got this difference between seed-coated cover crops, and on the right, you see the control of the cover crops. And we measured the biomass because we saw some differences. Actually, here you cannot really see the difference unless you're really experienced. And we thought like, okay, well, we might want to measure it, and we didn't have GPS or whatever, so we had to randomize the how do you call it? We had to randomize the samples. So we just threw a frisbee randomly and took like 1 square meter, and but it's randomized. I mean, unless the guy is really skilled in frisbees, but he wasn't. So yeah, it was definitely randomized. And we got those beautiful statistics too. That's the thing. Like you really want significant statistics. And I mean, come on, with those differences, we basically doubled the biomass. I mean, my mind was blown, and I'm the first one to be surprised, to be honest. But what does it mean if we double the biomass? Well, it means that we actually double the total forage nutrition. So the farmer actually put some pigs in there, and they were foraging on those plants, and we got what, 75% more protein in there. We got 127% more cellulose and 109% more ashes. So that would be the minerals. And we didn't measure the below ground biomass, but I would guess that you would have more roots and actually more root exudates, and so more carbon in the soil. So super interesting. And also, like we did a seed coating. I think it was 6 liters of compost per 100 kilograms of seeds. So it's quite cheap when you think about it. And here it's a picture of this farmer that used the same cover crops, but just a bit later. He just had something left and he just put it in his field. And I hope you can see the difference with I don't know, Brian, do you see a difference?
I see a difference
Yeah, I see difference,
on the
definitely.
right.
Or on the right hand side, you can definitely see there's
Yeah.
more foliage.
Yeah. And I sent it to to David Johnson. I thought like, hey, dude, you gotta see this. I followed what you were saying, because I got those guys that I never met, but I'm just admiring so much, and I thought like I'm gonna reach out to him, and I did. and he thought it was very funny, the story of the frisbee, because I told him. And yeah, and then he mentioned it in his presentations, and I gotta be honest, guys, like really, that was a good day for me. I don't know, it's just it was great. And yeah, so we tried it again in with wait, how do you call it? Barley in 2021. And also, I don't only work with organic people, actually. I think conventional has a lot of room to grow. And if you're in direct seeding, it's just so different. Just a side note here, but I have this farmer called Géraud Dumont in Belgium, who's doing an amazing job. And you see the difference between, okay, he's in direct seeding, he's using glyphosate. I think he's at 2, 3 liters per hectare. And yeah, basically making sure no weeds come up and direct seeding, and you have this neighbor that's organic. And my God, what a difference. The soil is completely different. When it rains, he says it's like this. He said, I receive water from my neighbor. I guess you know what I mean with this. So I think we really need to go beyond this organic versus conventional. I think there's so many farming systems. And also, I saw some fungi there. Like he's using glyphosate, and there's this fungi coming up, and just really interesting. So just a side note. So I work with this farmer who's in conventional and direct seeder, and we tried doing some seed coating with barley, and we did not do enough reps. So meaning we did 2 strips of coated seeds and 2 strips of control, and we still could measure a difference of approximately 4.7% of yield increase. But I didn't get those sweet statistics because we didn't have enough reps. But interestingly enough, we got the same amount for those 2 strips. It was the exact same amount of yield. So I cannot say for sure scientifically we had a difference, but it's really close. And I think the picture on the right really shows you the structure, and the rhizosheets on the left, which was the barley that was coated compared to the control, was quite impressive. Mm. Yeah. Okay, so how does the root system look for control and coated seeds? And on the left, you see the control, and you see a bit less of this rhizosheets, those sweet rhizosheets we all want. And on the right, in the middle, you see really those rhizosheets. And I always take this picture in my presentations because I really want to show people, like, look, this is just a picture, and it depends on who sampled what and which plant and blah blah blah. So I, on purpose, I put a picture with smaller roots because the smart people here would say, well, it has shorter roots, so it's not that good. Well, the roots are still in the soil. I mean, now they're eaten, but we couldn't take out those roots. So the middle picture has shorter roots, but it's really because we just took it out and it just stayed in the soil. So really focus on this exudation slash microbial community building around those roots. And on the right, you can see this difference. Yeah, it just speaks for itself. Oh, just a side note also, like sometimes if you do some seed coatings or trials, experiments, sometimes the data is not where you think you should look. So just a warning, or I just want to help people if they're going to do experiments. We had these experiments with barley that I just talked about. Well, the next in October, so during the season with barley, we actually had this underseeding of clover, and the clover was growing was growing much less than in the control. And I said to the farmer, God damn, I invented a seed coating that makes clover grow less well. And he told me, no, dude, it's just probably that the clover did a bigger amount of biomass. It probably produced more biomass, which could explain the higher yield. And also because of light competition, you might have slowed down the clover. So get yourself a really good farmer, because I'm really a student. When he talks, I listen. I have no idea. I'm not a farmer, and they know so much. So just saying, sometimes you gotta, yeah, look where you didn't expect to look. So 2022, 2023, we did some trials with wheat, and we did some plant sap analysis. So listening to John Kempf, etc., we just thought like, okay, this is really interesting. For people that don't know, plant sap analysis is comparable to a blood analysis. You take some blood and you say like, look, um, how much iron do I have? How much ammonium do I have? I mean, we wouldn't have ammonium, but you understand for plants, it would be the blood of the plants that you analyze and not really the tissues. It's a different thing. A tissue analysis is different, but it's more like what is circulating inside of the plant sap. So this way you can understand like how plants function and what deficiencies are there, etc., etc. And to our surprise, we we did a lot of treatments, but I really narrowed it down also because of our budget. I we analyzed control, compost seed coating plus compost extract, and seed coating, compost extract, fermented comfrey extract, humic acids, and molasses. The last one is the treatment that the farmer wanted. And because he was saying like, look, okay, you want to separate everything, but just put everything in there. Like just put everything in there and we'll see. And I thought it was a good idea, so we just did. And as you can see on the left, you have this 0.2 level of sugar, so percentage of sugar. That would be the control. In the middle and on the right, you have this biologically treated wheat, either with composite coating and extract or else. And we got 50% more sugar. So that's quite puzzling because we thought, okay, well, 50% sugar, that's actually not bad. And when you think about it, a plant is a solar panel. What is it doing? It's transforming solar energy into chemical energy. So from solar energy to sugars, basically. Now the question is, what if I told you your energy plant, I'm gonna make it 50% more efficient? Come on. Like the thermic motor of a car is 30% conversion rate. So you put 100% of petrol energy, chemical energy in the form of gasoline, you get 30% of kinetic energy in the form of movement. That's the conversion ratio. And we got more than, I mean, plus 50%. So insane. Nitrates. We got a reduction in nitrates by 45 till 85%. Disclaimer though, 12 ppm of nitrates is not a lot. So it's a big drop, but it's a big drop of not so much. So gotta stay scientific. However, ammonium was quite high. We got 2,500 plus, and we got a reduction of 33% of ammonium in plant sap. And when you listen to those guys called John Kempf, AEA, etc., which are miles ahead, or they really changed the way I view agriculture, and probably a lot of people too, they are mentioning, okay, well, insects are looking at plants in infrared. They can see which plant has which amount of nutrients and which one they want to attack. That could explain so much why different fields with different treatments get attacked and the other ones not. It's just they view the plants differently. And when you reduce ammonium, or at least liquid nitrogen in plant sap, you have less stinging and sucking insects. So we're talking about something that put more sugar in the plant sap and also reduce pest intensity. So really interesting. But I thought also like, oh, maybe I have less nitrogen in my crop, which would be a bit annoying. Like wheat, you want to have 12% protein, for example. Well, the total nitrogen was constant. So that was really puzzling. How can we have the same amount of total nitrogen and less liquid or dissolved nitrogen? Well, let me introduce you to the health pyramid of the plant health pyramid. I hope everybody has heard about it. If you didn't, I'm gonna explain it really. I'm just gonna explain it. Okay, so according to John Kempf at AEA, you got complete photosynthesis. That would be the basis. If you want to really make the best of your plants, you have to enhance photosynthesis. You also have to, the next level would be complete protein synthesis. protein synthesis. And after that, you get increased lipid synthesis that would protect the plant with its cuticle, etc. And in the end, top level, high level stuff, you get plant secondary metabolite synthesis. And that's when the plant starts to defend itself with chemicals. I mean, plants are the best chemical makers ever, and they defend themselves against pathogens. So what did we have? We have the constant amount of nitrogen, but we have a reduction in ammonium. So I'm hypothesizing here that we produced more protein because in total nitrogen, you're measuring everything inside the plant. So that wouldn't be actually a plant sap analysis. Total nitrogen is kjelldal, so it's the whole plant. So we get total nitrogen and less soluble ammonium. So really interesting. But that's just one data point. So you got to measure in later in the season, and we saw the same thing or even more. We got 130% more sugar and 150% more sugar in plant sap. And interestingly, also, we got more manganese in the plant sap. And not just a bit, 62%, 78%. And we didn't add manganese, I can tell you that. And the thing is, manganese, if you're gonna really nerd about it, manganese is a cofactor to an enzyme in the photosynthesis system. So an enzyme is a protein that's going to make a reaction possible. So without an enzyme, some reactions just don't happen. And you have to see the cofactor of an enzyme just like the little screw inside of a machine that's going to make the machine work. So here you get a small example of what a enzyme can do. It's going to reduce the activation energy on some chemical reaction. So you got a log of wood, and you know it's not going to combust out of itself. You need to activate it with a match, and then it's going to burn down, and you're going to release so much energy. So that's the graph going down. It's a release of energy, but you got to give a bit of energy so that the energy gets released. Well, same thing with an enzyme. An enzyme is going to reduce the amount of energy needed to get to this result. And you got this enzyme and the cofactor, that would be the little screw. And mostly in plants, it's complex metals or actually quite rare metals, molybdenum, manganese, copper, etc. And they get it via the microbes. So crazy that we get more manganese in our plant sap and also that we get more sugar. So I think they're correlated. Yeah, and so ammonium, we also saw the same trend. I didn't add nitrates because who cares? But ammonium, we saw the same decrease, and total nitrogen, again, the same constant amount of nitrogen. So that was such a cool tool to understand what we did. And now I'm asking the question, like, did we just hike 2 or more steps with just biology? Because remember, John Kempf is just talking about complete photosynthesis being dependent on biology. But the way I see it, or the way what the data seems to say, is that we got complete protein synthesis, or at least a big part of it, happening just with biology. So yeah, just really cool to see that some guy on the other side of the ocean was talking about some stuff, and we just measure stuff, and wow, it's just there. So really exciting stuff. All right, and so I'd also tried with tomatoes. Let me look at the time. I'm good. All right, so we tried with tomatoes because I felt like, okay, we gotta help those market gardeners also, like, and we tried with different varieties. We took Rio Grande, San Marzano, and Roma VF. And San Marzano is the tomato that the Napolitan pizza makers use to make those beautiful tomato sauces. So it's a really important crop. And I said to this, yeah, those are people in Germany. They're really, really nice. Elsa Jean-Jean did this study for me, and she was super excited about the workshops I was giving, and she wanted to try it out herself. And so we did this this small protocol. It's not a very scientific protocol in the sense of we didn't have statistics, but I think you're gonna like the results. But it's called a screening. So a screening is called like, okay, you have no idea what you're doing and just gonna try something out. So that's what we did. So the control on the left here, you see, received 100% bark mulch. So that's something that's the farmer used, and that's also what I also always do is just use what you usually use. And they were also using 70 grams of Multiflow, which is a slow release fertilizer. And on the right, we use 50% bark mulch, so what they were using, and 50% micro-bridge compost, the compost I give them, and also, of course, 70 grams of Multiflora to make it even. Now, the question is, you want to show biology works or not, and so you have to make sure that the NPK levels of your compost compared to the bark mulch are similar. Otherwise, and logically, you could conclude that the differences are due to the different levels of fertilizer. So we checked the levels, they were similar, so let's go. And on the left, you can see the difference. So that was in those small pots. And I don't know if you see it on the screen, but it's quite crazy how different they look, but it doesn't stop here. So there was this stage, and then there's the repotting stage in P9 pots. And you can see this is the San Marzano variety. And here you get the control. So this is how they look like. They have a beautiful green, crazy. And on the right, you got this the ones treated with compost, which are much bigger, but they have this light green color, which make me think that they just were ready to be potted maybe a few weeks ago. But the size is just crazy. And at the stage of repotting, we put 10% of micro-rich compost just to be sure. And so it was time to put it in the field. We harvested the tomatoes. We only got 2 harvests because after that there was a huge hail, so ice cubes coming out of the out of the skies, so they just destroyed the complete crop. So I got the data of 2 harvests. And on the left, you can see the Rio Grande variety with compost on the left and the control on the right. In San Marzano, you got compost on the left, so the ones with micro-bridge compost on the right, left, and then control on the right. So that's the first harvest. Like, let's see the second one. Well, second one, quite similar. We got on the left this compost-treated tomatoes of the Rio Grande variety, and then the control on the right. And then San Marzano variety with compost treated and the control. So you can see quite a bit of a difference, and I like numbers, so I just calculated it, and we got 72 to 77% more harvests with compost. I mean, okay. But life is beautiful, but sometimes it gives you lemons. And life told me, not so fast, my boy. In full disclosure, we had a third variety, and this third variety wasn't feeling so well apparently, and I produced less with my super amazing micro-rich compost. Huh, right? We got 2 beautiful tomatoes for the first harvest, and the control got a measly 7 tomatoes. And the second harvest was, yeah, a bit better for the control again, yeah. But I would say, okay, well, of course it depends on the variety, but I got my hunch would be, okay, well, this variety actually had quite a bad time overall. When you look at the differences between harvests above, like they're producing much more. So I would say like, okay, well, this variety was not feeling well at all in the climatic conditions. So yeah, I'm just putting it out there so that basically full disclosure. So this is what happened, and these are the results. No point lying here. All right. And we did a trial again with legume mixes in Belgium. We coated the seeds also. We put 4 control strips and 4 seed coating strips. Here you see the seeds that got coated. So we had fava beans, lentils, and veg. And after a few weeks, we got this emergence. So the seed coating was coming out faster. And yeah, let's see the results. So it's in March 2025. We're going to harvest in August. So let's see how this evolves. So work in progress. And I did a bit of a pot trial also with seed coating. And I wanted to share this with you guys because I was quite astonished also. I just put a bit of, yeah, just some pots together, took some farmer soil, and coated some seeds and some not. And yeah, we see a really big difference in root structure, and also how the roots respond to the soil and the coating, because you get this rhizosheets also in the coated parts of the barley. And also, latest trial, we did some trial with aubergines, and that was really nice. We tried some. I thought like, okay, this tomato thing was so interesting. I'm gonna try with also a solanaceae again, like a nightshade. And we tried reducing the amount of compost, right? I told you we were doing this, how do you call it, like screening. And so we started with 50%, but actually I thought like maybe we can reduce the stuff. And we tried with 5, with 0, with 5, with 10%, right? And so here on the left, you can see the 0% compost, how the aubergines look like, the 5%, and then the 10%. I think we can still see a bit of a difference between 5 and 10, but I mean, come on, between 0 and 5, I think it's quite obvious. And after 1 month, so really nice to see. And in May 2025, so that would be 1.5 months later, it was time to put it in bigger pots. And we can see that the control actually caught up a bit. We see that it has some nice leaves, but I mean the leaves on the 10% are just quite massive. So yeah, the question is like, what is the right proportion? Well, we're still looking for it. So they're going in the field this year too. So we're gonna have some yield data. And this was a stage when we repotted it, so you can see the root systems also. Yeah, looks like a nice bunch of of beautiful roots in the coated seeds, or no, sorry, in the substrate with compost. And you can see also the plants are a bit bigger though. So, and now the question is like, okay, well, I created this company called Resolutions, and I wanted to say like, why did I start this stuff? Well, we need to regenerate our soils, and it's quite urgent. We gotta be fast. We don't have that much time, and the way I see it might be wrong, but the way I see it, we need good compost, good microbes, and we also need a transfer of knowledge. We need a lot of people to get on board, and they don't have time to read all the scientific literature. We just help them to understand what the science already knows, because you don't have to invent anything. That the scientific literature, when I look at it, it's just mind-blowing how much we know. But the problem is we don't have scientists, or we don't have that many scientists talking to ordinary people, the plebs like us. And so we need also a translator, because it's really difficult for farmers to understand what the scientist is blabbering about. And we also need to build communities. We need to exchange informations. We need to help each other to grow further, because we could stay in our own backyard, but it's not going to help so many people. So I started producing compost. To be honest, I prefer you make your own. Just make your own compost. But if you don't have time, etc., well, you can hop on our website and you can buy some compost. Right now, we're unfortunately sold out, but we'll have a new batch coming in February. And if you want to do some trials, because again, we need science, we need to show scientists with statistics, etc., that it works. And so it's really important to get more data in this field of living and microbial compost. So I got the strategic reserve. I got this reserve, my own little stash that I'm very happy to send. But only if we can get some really cool data, and the setup is cool, and we get some statistics, etc. So we can make this movement, or at least this technology, move forward with some scientific data. I also organize workshops, and I focus on theory and practice. So I always start with the basics, and for me, there's no way to go around this. It's about soil structure. Once you understand soil structure, it goes into soil biology, it goes into microbridge composting and the applications, whatever you can do. But really, you gotta understand how a soil works. So I always start with this. You can do a bit of microscopy or redox, that's really interesting, but soil structure all the way. So this is what I did some 30 plus workshops over the years and keynote speeches, and it's so rewarding. I just get this farmers looking at the microscope for the first time and doing this piles. I mean, it's just I just love to do this. Oh, and by the way, Brian and Keisha and Rayleigh, if you could just help me out on that one. You see the picture here on the right? Do you see
Yeah.
my mouse?
Yeah, I
Yeah,
see it.
we called it the potato, and to be
The
honest, I have
potato.
no idea what it is. It's so big. That's it. It's times 40 magnification, and it's huge.
What if
Like
it's
I don't know what it
an
is.
egg, like
Oh,
insect egg or something like that?
huh.
Keisha, any thoughts?
I wish I could focus up and down on it.
All right, yeah, go through it, right? The focal
Do you have a
plane.
closer picture
Yeah,
of it? Did you take anything with a microscope that I could?
Oh, I'm gonna send it to you later.
Yeah, do if there's
yeah.
But I thought like, what is this? And I thought I have to include this because maybe somebody in the chat has
But
seen this
no.
before.
The
But
egg.
And
well, that's huge. I mean,
the
that's a
egg.
40x, so
Yeah.
that's big. That's really big.
Yeah, exactly.
It's kind of
And
smooth and it's kind of regular shaped. Yeah, I don't know. It'd be interesting to see if you got something that goes through all the focal plane of it.
I need to give you a better image. Yeah, I'm
Yeah,
not helping you guys.
yeah.
But yeah, we had a lot of fun. We just called it a potato and moved on. Also, if you ever meet someone teaching you stuff, it's such a good sign if people say, "Hey, I just don't know." Like this is such a important thing. You have to, yeah, you need to have people that are comfortable saying, "Hey, I have no idea."
Right, right, right.
And this is how you can move on, right?
Yeah.
Um, and also, I wanted to tell people that speak German, so Switzerland, Austria, and Namibia, maybe. You can watch on YouTube a free video about the microbiome symposium that I did. It explains a bit more about cell structure, etc. And big shout out to Stefan Schwarzer. I hope he's there and listening. Who's doing an amazing job with Aufbau in the Landwirtschaft. He's doing a really important work organizing those symposiums about the microbiome. So lots of love to him and his team. And yeah, and so now we're building up this platform of courses. It's not online yet, but basically I'm trying to get all this literature that's popping up in my mind, and I'm just like getting this all and getting this into some understandable format for farmers. And that's the way it looks like. We got some slides, soil structure, soil microscopy, etc. Redox, viruses, also really a crazy amount of information. And if you're interested in this, it's fine. But please, for the love of God, don't buy my courses, but get yourself a community. Like really, just exchange about this stuff. Like you have no idea how different people work and they have such crazy minds. I mean, some people are going to post every day. I think people know who I'm talking about. Uh, and uh, and yeah, it's just so rewarding to be able to exchange about yeah, what do we do, what we can do, yeah, just to yeah, build yourself a community. It's so important, and yeah, so I got super lucky to meet those people, and I thank them very much every day. So that will be the conclusion of my presentation. I think I'm on time. And to get a bit of marketing in there, you can scroll or you can go on my website and yeah, just subscribe to my newsletter, and I will know if you're subscribed in the last 10 days, and I'll give you 20% off on your next purchase. So that will be it, guys. Thank you so much.
Awesome
All
presentation, Juan.
right. Thank you, Juan.
Yes,
Appreciate it.
it's
fantastic. I love your slides, by the way. I'm a slide
Thank
jockey
you.
and I always appreciate good work. That was fantastic.
you like the one where it was
Thank you so
climbing,
much.
right? Up the pyramid. Brian
Oh
was
yeah,
Oh,
yeah,
yeah.
he
exactly.
loved Brian was like, oh yeah.
Awesome. When we cut the screen share real quick.
Yeah.
Yeah, Juan, that was fantastic. It just the effort that it takes to conduct a trial cannot be underestimated. It is such a rare, precious thing to be able to show the public. And so we really respect you being able to do that and to share that with us all. So,
Yeah, thank
this
you so
is
much.
For
massive
sure.
respect.
For
I
sure.
actually thought I was going to make a list of everything that went wrong. Like you see a part
Yeah.
of it, but
Yeah.
like, guys, like let me tell you, there's so many stuff. It's hilarious how everything that can go wrong will go wrong. So yeah,
Yeah. You
thank
know
you
what?
so much for having me.
And I'm glad you
Mhm.
didn't, don't shy away from that, right? It's so important to be able to share to people successes, but then also failures. I mean, failure is
Mm,
part of the process. And,
yeah.
and just to make it like, oh, everything's so great, and then hide what
Mm,
actually failed is just a disservice to everybody
exactly.
because people are going to do this work and go like, oh man, why? He did so
Yeah,
good, and I can't, make it to work exactly like he did, and that's just unfair, right? So, yeah, transparency is a
yeah, I
key thing.
agree.
Yep.
Yeah,
For
that's why
sure.
I showed the tomatoes. Like I was so surprised. Like, come on, guys. Just when you think you got it, that's the story of my life. Just when you think you got it, nah.
Proves none of it works.
Yeah.
Oh,
And there were
no.
some good comments in the
Yeah,
chat, around especially like the tomato thing. And potentially it could be around the species that species could have been,
yeah,
grown, developed over time that is in a non-microbial kind of function, and it actually just functions better in that agrochemical kind of space. And I think that's probably a very
yeah.
true, or could be a
Hmm.
very true statement, right?
Absolutely.
Yeah.
Cool. Well, we got some really good questions here. So at first there was three, and then all of a sudden, boop, 14. But like always, would love to have people raise their hand and ask their question live. So if you ask a question in the chat box, use the little raise hand function and we can unmute you. There we go. And then we can get to some type questions. So we have Katrine de Graaf here. Hey, Katrine. Hello, hello. Looks like she's. Oh, wait.
I was muted. I'm
There
sorry.
we
Yes,
go.
There you go.
there you go. Yes, my question. In France, Ardèche, and I'm a soil food web consultant graduate, and I'm currently writing a business proposal to start a composting facility, composting business here. If there is one thing that you would want to tell me or recommend me, or one advice, what would it be?
It would be huh. So I'm making compost myself too, and I will tell you the biggest advice, or at least the stuff that I learned, is unlearn the stuff that you think you know. Because just to give you an example, in the Johnson Sioux community, you would have people saying, just as an example, oh, the walls need to breathe, right? It needs to be a breathable
Mm,
wall. So it's going to put a constraint on your operation. So you're going to go with this belief and say, okay, well, I need to have a breathable wall. Well, did you test it? That's you cannot test everything, right? But just
mhm.
make sure that what you think you know might not be true, and
Yes.
you'll avoid, I hope, a lot of mistakes. But you can also give me a call. I'm happy to help further if you have more details.
Thank you. When I'm a bit further in the process, I'll probably
Mhm.
take you up
Sure.
on that one. Thank you, Juan.
Hmm.
Great. Thanks, Katrine. Appreciate having you on. All right. So let's do some type questions here. I guess first one, quick one. What are the seeds coated with? I might have missed it. Of course, these are
Oh yeah.
extract, but is there anything else besides the
Hmm.
extract?
Yeah, I apologize to the audience because I didn't have really time to explain the seed coating. But basically, I use more compost than Johnson because I'm really paranoid and I just want to make sure there's enough compost when I'm doing a trial. I'm using approximately 6 liters of compost per 100 kilograms of seeds. Depends a lot on the seeds, depends on the surface actually. I like it to be really black and that it yeah, sticks to the seeds. I also make sure that I drill it, that I seed it quite fast because you're running against time. You have this compost that's really moist, and then you have the seed that's going to dry out or absorb the water. So also a misconception is that people think, okay, well, the seed is going to dry out. I have to put it on a how do you call it, like a tarp. But actually, it's not the seed drying up. It's actually the seed taking up water. Different thing. So you need less surface area. Let's say you can put it in a big bag. It's going to dry up. You might want to move it a bit. But to answer your question, I put a bit of molasses, so 6 liters of compost. I would say between maybe 15 milliliters of molasses and 125 milliliters of of milk. You mix the milk with the molasses. You can go on my YouTube channel. It's in German, but you will see you get a feel of what you need to do, and you add approximately the how do you call it? You have compost, but you also need to add water to it. So you have this paste, and make sure you don't make it too wet because you're going to coat your seeds and they're going to be too heavy. You're not going to be able to to see them in the how do you call it?
Seed
Cedar.
drill.
Yeah. And if you're going to store them and you put too much water, they're going to germinate. So that's less nice. So there's a bit of trial and error, I would say. What else can I tell you? Ah, yeah. Also, don't forget to regulate the seeder. So, if you're gonna coat seeds, don't forget you put, depending on how much you put, of course, but you put more weight in it, right? So you put more water in it, you put more compost in it, and if you're gonna compare it to a control, if you have 100 kilograms of seeds and you put 100 kilograms of seeds that are coated, then you have, wait, 6 liters of compost more, probably 4 liters of water. You have a bit of more weight, so you have to adjust the seeder. Plus, make sure that the seeds slide very well in the seeder. And as I'm saying this, please note that it's all mistakes that I made. This is this is why I'm saying this, so that nobody gets a bad experience and we can all move as a community. So yeah, check the seeding rate, and make sure it's dry enough that it slides through the seeder
Hey,
and
Juan,
make replications.
can I ask a
Yeah.
follow-up question on that? So on the seed drills,
Uh-huh.
you found that, different manufacturers perform differently with the coated seeds than others as far as, the ability for damming and kind of blockages and so forth, or you found that once your kind of seed coating process seemed to work with most of the seed drills that are out there.
I would say yes. Really depends on the kind of seeds. For barley, I never had a problem. For wheat, also no problem. We had a small issue with bigger seeds, such as faba
Lavas.
beans, that actually got a bit more squishy. So I'll have to reduce my water content because it actually caused a few problems. Or yeah, it's trial and error, and we're really into this infancy. I forgot to say it in the presentation, but please take it with a grain of salt. These are my results, but it's also we're early. We don't know exactly how everything works, and yeah, remain skeptical, please. That's also what I would like you to do. Yeah.
Fair enough. So here's a question from Jeremy Petrie is, can microbes be used to break down glyphosate? I think I heard something
Hmm.
like that in a webinar a while back, but not sure.
Yeah, the only thing I know about it is that Johnson was talking about this, saying, "Hey, we found this microbe in my compost that, by the way, breaks down glyphosate." And I don't think glyphosate is the biggest problem, mostly the breakdown products of glyphosate. So I don't, out of my memory, I say AMPA, but I have no idea how it's called really. But that would be the thing to focus on. Like glyphosate breaks down, but the products are the problem actually. Yeah.
Yeah, no, we had some webinars with Elaine where she mentioned that like enough of a robust biological community can help break that down eventually. Well, we
Hmm.
can refer people to that. I think we did that
Yeah.
like
I mean, there are some forever chemicals too, which blows my mind too in a bad way. Like those Tefal the stuff to cook, those stuff. It just doesn't break
PFAS.
down at all. So I like the idea of microbes being able to do everything, but I would love to see some data. Um, yeah.
cool.
But glyphosate is really the most researched herbicide, so it's really getting screened a lot. Like when you look at what we used before, like dicamba, with like so many other really horrible stuff. Like if you can get a direct seeder and a bit of glyphosate, man, I mean, yeah.
All right. So Jeremy Petrie asking, is anyone on the panel familiar with the Jerry Giuseppe spice compost method and no turn, no aeration compost?
Hmm. I
Yeah,
don't.
we had a Maria. I can send you the webinar, Jeremy. I'll get the link and post it. In the Water Stories webinar, um, one of the water and soil consultants, Maria, she had a presentation about spice composting. It's basically just It's very interesting. It's compost. It's like static composting, but you're covering it with a tarp and
Oh yeah.
continuously watering it. So I'll try to get that link and I'll post it below where she talks about it. But yeah, it was interesting. It was the first time hearing about that because you had hear about Jonathan Su, you hear about static composting, you even hear about hugel culture is composting. But spice
Well,
kind of was new to me.
Uh, I hear you speaking German with Hugo culture, so I have to jump in.
Oh yeah.
It sounds a lot like what Walter Witte is doing. Witte composting, which is quite famous in Germany, where you actually cover the compost pile with a tarp, and you go. I mean, they say anoxic and not anaerobic. I'm still trying to figure out the difference, but you get a completely different. You get a fermentation basically. And you get some what we call bad guys, let's say, but more yeah, definitely how do you call them? Yeah, those anaerobic bacteria and then protozoa and fungi. But apparently also really interesting in the CO2 balance, you have much more CO2 loss, much less CO2 loss. And if you use this stuff on your field, you might also get all those anaerobes and facultatives, and you get the CO2 production because it's going to oxidize at one point right below your plants. So you get a CO2 uh, fertilization in a way. So it's interesting.
Yeah, that was one really cool thing about hugel culture. Learning that was like from Sepp, it was basically it wasn't just a way to grow plants. It was also the main function was composting. So it's just leaving there for a long time, then harvesting it years later to harvest that compost instead of just using it as, the grow like the plant bed.
Mhm.
Okay. All right. Next question from Arif. In the tomato trials in general, how do you decide what percent of compost to add at stages of the experiment?
Mm. Ah, is this person referring to the tomato experiment?
Yeah, the
Yeah.
tomato
Ah,
trial.
yeah. Yeah, as I said, screening. So, in short, no idea. You gotta start somewhere. And
Right.
50% sound like a good thing. And now I'm at 5%, 10%, and I'm gonna try 3%. So,
Yeah, I think that's a little bit like on David Johnson's side of the house, right? Where it's the minimal amounts can still gain huge, because once the plant makes a connection with the microbial community, off it goes type of thing. So I'm glad that
mm,
you're actually doing that kind of work of really kind of fine tuning kind of what the percentage is in there. Because the inoculum grade compost is expensive when it comes down to it. There's no way around it. And so whatever you can find to cut
mm.
maximum benefit, least amount of cost, I think that's great.
Yeah, it's hard to figure out what would be the right market value because if I had to count the hours and stuff in the making, I'm not even talking about R&D. It's I mean we're early, so this is why it's expensive. I mean relatively expensive. I think if your costs are less than how much you produce more, you're good. But of course, it would be great if it would be incredibly cheap and people can just run with it. Yeah.
Wow, catalyst compost is so valuable.
Well, I
Yeah.
mean, I think, Keisha, we had this conversation too around, if it came down to if you price it per the amount of microbes, then an inoculum grade could be really valuable, right, compared to the cheap
Mm,
bulk compost. But if you're just looking at volume and that's it, yeah, the inoculum grade stuff is going to be way more expensive, but the
mm,
biological value is so much higher that it really is a good
mm,
deal.
I think for years and years and years, it's been just not really broadcasted properly why it's so expensive.
Yeah.
Um, Often you'll hear it said like it's so much better than other composts that you can charge 10 times
mm,
as much money. That was definitely the advertising technique that got me into making the product. But the truth is typical composters get paid when they receive waste. Most of the compost, almost all the compost on the planet is a waste product. It's not made as an inoculum. It's almost like food. You could compare McDonald's and, a five-star restaurant. That's the difference. So when we make compost, even though we have giant tractors and trailers, um, it costs us, $350 a yard plus. Because once you get these microbes, you have to water them. You're doing microscopes. You're every single bit of care is taken. The covers even, that's not typical to put covers on compost. So even keeping a beautiful, clean lot, that's just not typical. So when you get into large scale versus what we do, there's just no, it's hard to compare it. And then when you're talking about waiting 12 months to get your product, of course one's sold out. Like you're always going to be sold out unless you can, how many bioreactors are you going to need to make to service the area you're working on? So, I mean, I love the Johnson Sioux. It makes epic compost, but it's like, yeah, what price do you charge for it? A lot,
Yeah.
like more, way more than I do. That's for sure. What, like 2,000, 2,500 US per yard at least, if not more. Um, I can't imagine.
Right. But I'd love to have like there's a metric like the cost per microbe or cost per, something in that sense, because then it's like,
Cost per
oh,
microbe.
Well, it makes sense. I
Yeah.
mean, there's a cost when you're looking at chemicals. How much does it cost
sure.
to produce the chemicals they use? Like they're not being transparent with that number yet. It's, after there's enough large scale trials, I think we will be able to put prices on the microbes. I mean, if you
Yeah.
look at our Casbar trials, Brian,
Mhm.
um, year after year, I've watched him drop off every other product but ours. Why? Because on his own, he's like, I'm buying protozoa basically.
Yeah.
Like, I'm here for the protozoa. The other compost has none. So, he's slowly, our compost is really expensive compared to everything else. And I've,
Right.
I've watched it. He's just cut everything away because they're doing the data. They're running numbers. And until people start sharing, thank you for sharing your trial.
Exactly.
It's really hard for us to get farmers to talk about it.
Yeah.
Nice.
Fantastic.
Well, here's an interesting one. I mean, this is a pretty general question, but is there a way to fast forward the composting process? Like, what is the fastest? It seems like that question is what is the fastest way to making very high quality microbial compost or microbe-rich
Thermophilic
compost?
compost is the fastest way.
Yeah.
It's the fastest way. You can have, I mean, on my very best windrow, like very best windrow, we got it through its minimum heat thresholds that we had to for the state in 15 days, had it down to temperature within a month. So 30 days, and it's above its microbial minimums at 14 days. So the microbes are there. It's just a matter of waiting for it to, finish working itself out. But a lot, you look at large scale compost, they're going to heat that stuff up 14 days and then sell it. That's why you get it hot off the truck all the time.
Yeah.
But you can, like, I mean, if your
Yeah.
recipe's perfect, if you do like the slow and low, I call it, your temperatures aren't getting up above 150, so you're still getting great microbial diversity, you're still getting all your protozoa, and it could happen in as fast as a month, but more typically 2 months, which is fast compared to a year. Um, that being said, I just did like a static pile with my food waste, and it's so insane in the microscope. I was like, wow, this compost is blowing my thermophilic stuff out of the water. It's like I've been looking at it under the microscope just for fun because it's so crazy diverse.
Right.
There's a lot we don't know about growing microbes.
For sure. So a bit more trial questions here. It seems like there's a lot of like logistical trial questions.
Cool.
Um, so Citros is asking, how often do you add compost and how much in terms of quantity per each stage of an experiment or a trial?
Well,
Like, like at the
we
seed coat and kind of like what stages are you applying
yeah
compost and extract?
for the we actually start much more at the start. So the idea being the rhizosphere and the roots come out. We want this biology right away. In our wheat trial, we added some compost tea to all the treatments. But mostly it's really, unless specified, actually only at the start. So when I said seed coating for the barley, for example, it was just seed coating. For the tomatoes, I specified 50% at the beginning, 10% when we added more pots. I mean, when we moved them into pots, then it was 10%. And for the aubergines, it was just from the start. So I think the less you have to do, the better. I mean, depends on how much it costs, right? But every intervention is has a cost. So, mhm.
A question for you too, Juan. So,
Mhm.
compost, obviously in the form you've been going through the seed coats as well as the, in your mixes. How much
Yeah.
in these trials are you doing any kind of liquid amendment, like extract adding after in ground kind of situations?
I only did it with the wheat trial where we did compost seed coating plus extracts.
Right.
And also a really cool point also is, okay, you want to put biology in the soil, but really, if you want to have an impact and a long-lasting impact, it has to fit the farmer day-to-day or schedule or possibilities. And it seems to me that seed coating is a good way, and some farmers prefer extracts. So. Yeah, and I didn't show the whole data, but also because it wasn't significant, but we saw no difference in yields between extract and seed coating. So I cannot really tell you 100% statistically significant, but we saw the differences or the numbers, and they're really close. And also, what I also didn't tell, which I didn't have time to show everything, but also that it makes a huge difference to add nutrients to microbes. I mean, it was what everyone thinks, but we really saw a measured difference in, I think it was a difference of half a ton of wheat if we separated or if we just added microbes or we just added nutrients. And if we put those cells together, then we get this synergistic stacks of Kemp where we get this stuff that, yeah, that adds on, let's say.
Yeah, I think you hit on a point too, which is, when working, especially in agriculture, anytime that you have to use machinery, invoke a lot of labor, those are all
Mhm.
costs that farmers are going to be intensely
Yeah.
concerned about. So like in your example of using like seed coats, you can do that at a relatively cheap cost as far as preparing the seeds. The labor that the farmer's going to really spend is the actual seeding itself, right? The tractor and equipment, all that kind of stuff. And
Mm.
so we're always looking for ways to say, how can we maximize the return on our labor investments and so forth? Like when we use liquid amendments, a lot of times we try to use fertigation. We're going to put it out through the irrigation system, such that it reduces the amount of time and labor and so forth. So always looking at those kind of methods, I think is extraordinarily important. And
Mm.
if you put together something that's really a lot of touches, a lot of labor, it's going to be a hard sell for those farmers to look at because again, that's going to hit their
Mm.
bottom line.
Yeah, I mean, sustainability only with a good bottom line. Otherwise, it's not going to happen.
Right.
It's just a reality. Yeah. Hey, I'm seeing that I got some private messages too.
Great.
Uh,
Secret message.
yeah, secret messages. Yeah.
All right, well, here's a good one, Jeremy.
Wow.
Have
Yeah,
any of
go
the
ahead.
panelists inoculated a compost pile with EM? If so, what were the
Mm.
results?
Personally,
I
I have
didn't
not.
try it.
I've not
Mm.
used EM to inoculate. Keisha, have you?
No, I find like the pile gets EM in it anyway. When you look at DNA analysis, like typically you're going to see 1,000+ species of fungi, 1,000+ species of bacteria. So, um, you know, often those guys already show up. So I think the inoculation and the way I make compost comes from my input materials over time. So the way that I cultivate the materials that I'm using, we don't typically ever inoculate with anything that isn't made by ourselves. So. Um, but I'd be interested in hearing if anyone has that experience.
Yeah.
Um, Here we go. Here's an interesting one. So I know Todd Harrington's talked before about creating specific inoculants for plants by growing the plants targeted in compost. So Jeremy's asking, would growing AMF inoculant plants in a finished compost be a good way to get AMF into compost?
Yeah,
Yeah. By all means,
definitely.
that is a
Especially if you're
methodology.
bringing it from its wild environment where the spores are already present in its root zone. Yeah.
Yeah. I
Okay.
think that's the, you know, I don't want to say future craft composting, that kind of thing, but I think it is a technique, especially when you're talking about crops that are highly dependent upon like AMF. Having mother cultures or mother piles that have those spores that when you add them into your growing mediums or your seed coats or your extracts and things like that, then you have those kind of already pre-selected species of AMF that are going to help those plants. I think that is a technique for sure that will gain more popularity as time goes on.
Nice. I had a question. We had our workshop in Costa Rica, and we got our thermophilic compost through actually almost four turning cycles. And we were going to leave it with them, be like, okay, guys, it's up to you now. You have to do this maybe one more turn. But they kind of just weren't interested in that. Because they thought, we're in the tropics. We don't need this. We're doing this in tropic system where we don't need compost. But
Mm.
you're working with folks in Peru who actually saw the use in compost. Well, for people in the tropics and some tropic ag systems,
Mm.
why would they be using compost?
Oh, that's
Why go
such a
beyond
good
just chop
question.
and drop for them?
That's really, I'm so happy you asked this. That's also a question I asked myself. Look, what happens when you chop and drop? So you got this Ernst guy going on the streets and he's 90 years old. He's gonna cut a branch. It's gonna come down. It's gonna compost anyway. I don't know if you saw those videos that date from the 90s where he goes like, "Oh, look, this is my soil," and he goes like into I don't know, like 10 centimeters, 15 centimeters of biomass. It's just composting like it should be. Like this is the Johnson Sioux or composting. It's just not really, it's just nature. It's just nature doing its thing. And Johnson Sioux, they make sure there's enough oxygen and it doesn't go too high in temperature, just like in nature it would. Of course, you have some places in nature where would go up in temperature. But my point being, you put and you chop and drop some materials on the soil, you get an amazing compost. Plus, you get all those worms going in. You also get all those micro arthropods and stuff like that getting in there the moment your soil touched the compost. So why would you bother making compost? I would say if you're going to start a syntropic field, that would be interesting. If you want to have trees, well, hey, you know what? It would be cool to have some fungi in there. Coating the seeds for between the rows. Um, I mean, it's endless. It's really when you start it. I think it makes the most sense. But yeah, I mean, it's basically composting in the place, right? It's tropic agriculture. So,
I agree with that. I mean, to stand up for them a little bit, Finca Luna Nueva is gorgeous, microbially speaking. We did microscope adventures there, so we were taking samples from leaf surfaces, pieces of bark, little wood chips, dirt in the nook of trees, like just everywhere. And it was like every sample we took had nematodes, protozoa, fungi. It wasn't just the soil, like there was life everywhere. So for them, they didn't need to make more microbes unless they wanted to share them. And that's what I'd brought up was, if their goal at Finca Luna Nuevo was to help other farms get started, they have the inoculum right there, and it would be a good reason. But for them, their goal was to have the best farm to table resort in Costa Rica. So it made no sense for them to make compost. I agree with them. Too much work.
Yeah, of course. That's
yeah, it is work.
Oh, here's
And we got to save every energy we have, every energy input or every labor. It's money. It's time. It's just let's save as much as we can.
Yeah.
hmm. Well, here, Indira wants to ask a live question, so I'm gonna unmute her. Hey, Indira. Let's see if your microphone turns on here. Can you unmute your mic? Da-da-da-da-da-da-da. There we go. Hey, Indira. Oh, looks like your microphone's still muted there. I'm gonna give you a minute to activate your microphone. Well, also, folks, before we close the thing here, we also want to mention that Compost Academy, we're going to launch a new live 10-week program in just at the end of July. And we've had our first round of students who are just ending their last week. And epic stories and videos of people going through it. I've just been blown away by the amount of just activity I've seen. People building compost, people doing trials, people already teaching workshops just after like a few weeks. I'm going to share the screen, but it's just been phenomenal to see that and be a part of that. So we don't have the sign up for the next one yet. So I'm having a hard time sharing my screen here. Give me a moment.
And then, dear, just go ahead and hold
So
on for a second. Once Raleigh's done, then we'll jump into your question because I know you got your microphone working now.
There we go. So anyway, so if folks want to be on the start for our next 10 rounds, just hop on to Compost Academy website, sign up for the newsletter and we'll let people know. Like we'll especially join the wait list at the bottom, and then you'll get the first notification about when the wait list goes live. And we're gonna have start a series of webinars that's gonna lead up to that wait list. So hop on and join the wait list right now. And we're going to have a lot of cool things to share between now and when we start. So we're aiming to have this, uh, we're aiming to start the next round at the end of July. So there's a bit of time, and we're going to try to get an early bird offer up here soon. So hop on there and join the wait list now if you want to join our next 10-week program. Well, I think it's going to be 12 weeks, but
12 weeks, yeah.
12 weeks, yeah.
no, I was like, is it 10?
Yeah.
So,
I'm
12 weeks.
We
repeating.
lost
Sorry,
2 weeks. Oh my gosh.
Sorry, guys. That was 12 weeks. 12 weeks. 12 easy weeks. All
A little
right.
panic came across Keisha and I's face. Huh?
Let's,
What about Community Day? That's still in there, right?
You're right.
all right. Well, it looks like Indira couldn't get her microphone working, so let me hear. Okay,
She
here's.
said she had it activated.
Oh, yeah,
She's
she had it
there.
activated, but I still
Yeah,
couldn't
she
hear
did.
her. Indira, try talking one more time. In the meantime, here's a question from Dylan. How to make
She's
compost
there. She's
wand?
there.
Well, I can't hear her. Can you guys hear her?
Oh, it says
Yeah,
it.
I'm there.
Uh, I
Sorry.
don't I don't think I can hear her either. Yeah.
No,
Yeah.
I can't hear
Sorry,
her either.
Indira.
Yeah. Apologies. Okay, so here's a good recipe question. So Dylan's asking to make your compost one, which materials are you using? It seems like a good follow-up would be like, what kind of recipe are you following to make your compost?
You gotta know, Dylan is this guy. He wrote like he built a farm. It looks amazing. It's in Portugal. It's such a cool permaculture design. And to answer his question, I'm using every organic material possible. So I'm using wood, I'm using grass, I'm using what do you call it? Cow shit, cow dung. Also, sheep dung is quite good. Basically, you want this ruminants bacteria also. You want some protozoa, so I would definitely not shy on grass and straw and stuff like that. And so you get this mix. You can start, I would say, if you're really starting, you can go 50% carbon rich, 30%, 35% medium nitrogen, and then high nitrogen you can go to 10, 15%. But I'm actually having a call with Dylan, so he's a bit impatient. I have to call him next week.
He wants, he wants
This
it
guy,
now.
He wants to get a jump on
yeah,
it.
a really great guy. I hope he can post the website of his farm because he's an incredible market gardener, directly selling to clients and great guy. Hmm.
Awesome. All right. I guess this could be the last question. See how it goes.
Mhm.
Kushal is asking, how does vermicomposting compared to
Mhm.
traditional composting? It seems like
Good
a good like
question.
what context would you be using vermicompost versus Johnson Sioux or thermophilic compost?
Can I start on this one? But I would love your opinion on also. Uh,
Sure.
I've analyzed like I think 10 different providers, and it was really different how the microbiology looked like. In my what I found is it's quite poor in comparison with normal soil food web compost. But still, I think results are interesting. Got this whole hormones and stuff. So it's not because the biology doesn't seem there that it has no effect, right? So yeah, I'm still unsure about this. However, the Johnson Sioux is also a vermicompost, so the best of both worlds, I would say. Yeah,
Comparing when you're looking at worm compost, any kind of compost really, even Johnson Sioux, like just it being a Johnson Sioux doesn't make it fungally dominant or beautiful.
that's true.
There's processes for everything. So I've seen
Mhm.
just poor Johnson with,
Me too.
wow, really,
Yeah.
with people that made it with such heart and effort, but they just didn't, they messed it up. So I've seen vermicompost that's just incredible. I've
Mm,
never seen it at
yeah.
scale. Um,
Mm,
I've never seen it at scale when people are feeding mostly food waste and sugary ingredients. Typically, I see like bacterially dominated, but I've seen gorgeous vermicompost. So it
mm,
can be done. I think often it's like what you're saying too about your Johnson Stew is also a worm pile. So is our thermophilic
mm,
compost. So if it sits for more than like 3 or 4 months, it turns into vermicompost. If it sits for 2 years, it's vermicast, and it looks gorgeous under the microscope. But it's got structure.
mm.
So I think a lot of times when people are composting
Exactly.
worms, they don't use enough carbon and they don't use enough wood chip.
Mm,
Because you think like,
yeah.
how could a worm eat a wood chip? But they can actually. They use the microbes to take it apart. So if you're thinking
Mm,
about structure and you're thinking about diversity, I think no matter
mm.
what kind of compost you're working with, you can make microbes because you're mimicking a natural system. So,
Yeah, I totally agree. Just wanted to point out also the Johnson Su, where we always get out caught with these methods and gurus and perfect ways to do whatever. No, it's depends all the time. And I'm glad you also had your share of Johnson Su's that didn't turn. I mean, it's just a method, but really, I think something that most people overlook, so it's useful for the audience, is the structure of the material. So you really want this breathable structure inside. You're gonna put 100% cow dung. Well, guess what? It's not gonna breathe a lot. You're gonna put 100% of grass. It's gonna go anaerobic, even though you respected the golden rule, like of distances and remember where those rules come from. It comes because of observation and so put it into context, right? Don't see it as a next commandment. So yeah, yeah,
Yeah,
cool that you mentioned.
and I think too that, when it really comes down to it, having a processor technique is great, but in true, if you're really going to be good at making these things, you have to have an understanding of the system and how it works. And you have to have really good observational skills. And that could just be observational skills with your
Mm,
eyes or the microscope or whatever tools that you're going to use. But those three things combined, process maybe to start with, the understanding of the system, and then the observation, that's what truly is going to get you to that space that you're going to be able to make, something that is actually really beneficial, really, really good, high, very, very high quality. Yeah, so don't skip on those things. Sometimes it's so easy for, especially in the agrochemical world, they just follow formulas. It's a process. I follow a formula, but without understanding necessarily what's behind it. And in here, where no, we need to retrain you to really have a deeper knowledge because these are living systems. There's complexity behind it. That unlocking that understanding, I think, is a key to making these things work.
it's really incredible. One of our students, I mean, everybody is composting in a different way. We're all
Yeah.
across the board, but we have one
yeah.
person who lives in an apartment and she's using vermicompost in her kitchen. It's an indoor in her apartment, beautiful microbes. Like, it works for her scale and what she needs. And I'm so
Mm.
inspired by her tiny biodiverse worm bin that's just like in her kitchen. It's, you can
Right.
do this at literally any scale and just for houseplants if you feel it. So.
Mm.
And I've seen worms, they become the savior of my failed piles. I can't make it to that third term, then I'll let the worms do the
Yeah,
rest.
yeah. Yeah, that's a really cool thing. Also, times heals everything. Like the question is how long it takes, but it will be good compost at one point.
Mhm.
Yeah, for sure.
Yeah.
All right, folks. Well, here's one last little question here, and I guess this could be a good final question. Also, shout out to Hugh Johnson for being here.
Mm.
Was I don't know her relationship. Was she working with the Johnson Sue system or working with Dr.
Oh,
Johnson?
they got this crazy publication that came out. I think it came out a week ago or 2 weeks ago on
Oh, awesome.
PFJ.
Uh-huh.
I'm gonna try. I think Huichu gave me the link. I'm gonna put it
Oh,
in
awesome.
the chat, or maybe she
Yeah,
can do it.
she
Yeah.
had some great links she posted, so appreciate you
Yeah.
coming here. All right, I guess last question. From an anonymous attendee. Is using agriculture municipal waste with a bit of sorting. I guess I'm using agriculture municipal waste with a bit of sorting. Is it difficult to get high quality or diverse compost? Um,
You want me to answer this because
sure.
I
I'm
spoke the
like
whole
just the
evening?
phrasing of the question is different, but
Ah,
go for
okay.
it.
Oh, let me read it.
Uh, Did I get the question right that depending on the material that comes from, maybe not, oh wait, is using agricultural municipal waste with a bit of sorting. Uh-huh. Yeah, I mean, it's like making bread or beer or wine. It's a process. So of course the ingredients are important, but mostly it's about the guy making the wine, the beer, the bread. So it's the process that's important. Of course, you need the right ingredients, but it's both basically. Using agriculture municipal, actually, I think it's a contradiction. Agricultural waste is not municipal waste, but anyway. Any waste can be composted. It's a question of how do you make your mix. Hopefully you don't have antibiotics in there. Depends if you have a lot of grass seeds and stuff and you don't want to have wheat seeds in there. But basically, as long as you have enough air, humidity, and time, and you reach certain temperatures, if you're afraid of pathogenic bacteria, because for example, if you use this chicken manure that comes from I think in the US it's called Tyson or something like that. I don't know.
Mhm.
I
Yeah.
mean, I wouldn't trust it on my plants, so I'll definitely go extra high in temperatures. But yeah, air, humidity, and time, I would say.
We
Know
found
your
too, we work
source.
with some really large scale facilities they take in all waste. So they're
Oh.
the largest municipal composters in Los Angeles. And they have more control than you would think. So some of the ingredients I wouldn't even bother. You know, those green bins that in the US anyway, we have the green bins that sit out on the corner, and the amount of trash and just nonsense that's in those bins, I don't think it's worth it. But these super large scale facilities can sort and really preserve some of the better input materials. And so we've found that just with a slight alteration to their operating processes, that we can make great compost. And I don't do it the way that I make compost. It's much easier, much faster, but very useful and inexpensive compared to what we do. So I think municipal waste being turned into high quality compost is where we need to move to in the future.
Yeah, that's actually one of the weeks of our course was how to improve low quality compost with biological inoculum. And Citros had another question, just asked about a Compost Academy Live. And yes, the soil mentorship program is live. And if you join the Compost Academy when we next open it, you basically have access to four programs for the price of one. So you have the whole soil community group, which everyone's discussing everything that comes to their mind. Week to week, our discussions, our classes, resources. This is a really active, amazing community here where people are bouncing back, questions about trials and composting recipes and what's going on in the soul mentorship program. But also, you have access to a self-paced program where you do have presentations, you do have demonstration videos that are going to guide you how to like the process of building compost, and you're also going to have the biological primer. That's, of course, free program that everyone accesses to. So you have a lot of components to this if you join the Compost Academy Live. So you're going to get your 12-week live mentorship program along with the self-paced program, along with the primer and the community group. So, and the people it's been so fun launching this and seeing it build and seeing it live. With the most satisfying thing is seeing the people start to build their own compost, people start to build their own workshops, and people start to apply it on their own farms. That's where it's really, really getting exciting for us because this is the first one we did, and yet people are already doing things in their community, which is immensely satisfying.
It's really fun, actually. We have our agenda, our 12 weeks of the things we want to teach, but the stuff that comes up during the week is my favorite.
Right.
Like chasing white papers and calling people that I haven't talked to in years, just like, oh, we got somebody with a really great question. It drives my education more than anything.
No doubt. I mean, really, the
Selfish.
questions push us in the areas. They're like, I kind of have an idea, but I really need to go find out the nuts and bolts behind this, right? And for sure, it's great.
Oh yeah, and we've just decided we're keeping everybody involved forever. So the students from last time are going to be on this
Yeah.
time, and the people that come on this time get to stay on. So
See the previous
It's turning
questions.
into a
Yeah,
big crazy
yeah,
group. It's fun.
yeah,
I
yeah.
know it's
Community.
a
Well, Juan brought that up, right? Community is one of the, I think, the most driving
mm,
factors of making this successful. And yeah, that's what we strive to do, is try to make community. You
yeah,
have to be
without
out
a
there
doubt.
alone.
That's why we all loved, like, everyone's kind of like retreat, retreat, get the next retreat.
You know,
Everybody go to
guys,
Vacation.
retreat.
what I really came up with is like, imagine there's a guy in a group and you don't like him that much, for whatever reason, probably because he's reflecting stuff that you don't like about yourself, right? But I'm just thinking like, hey, you know what? People are like microbes, They all have their function,
Sure.
might be a bit pathogenic, but hey, he's there and he's useful. Like he
Right.
does some great things and he's making it go forward. So just having this patience and tolerance towards different opinions or ways of functioning is quite beneficial sometimes.
Well said.
Yeah.
Yeah.
And no
Mhm.
one wants to do this in a silo. Like no one wants to just be the one person doing it. You feel like you're going crazy, and that just getting this community together helps you feel like, oh my gosh, there's other people that think like me, that care about the same things I care about, and it's incredibly special to have that. All right. All right, everybody. Well, I think we might end it here. Quick thing, Kushal says, have you considered collaborating with the Save Soil movement? It looks like a really great movement. We'll look into it.
That's
Um,
Sadhguru.
yeah,
It's, he's
Sadhguru, yeah.
he's a great guy. I was
Yeah.
I got lucky enough to meet him and I made him laugh. I don't remember what I
Oh,
said, but
sweet.
maybe he politely laughed. I don't know, but really great presence, really interesting guy. Um, but hey, I would just wanted to thank you guys for inviting me to show this data. Really, it's such a pleasure and honor to be here to be able to show this. And then, yeah, thank you so much. And I hope everybody enjoyed it. And yeah, just let's keep composting, eh?
Right, right.
Yeah.
Yeah.
Pleasure and honor having you
Thank you.
on,
Yeah,
Juan. Thank you
exactly.
for your
Thank
epic
you.
work. And yeah, just a quick shout out for everybody. You're going to get a replay in your email. And on the 19th, we have Matthew Gates. So he's going to talk about how do you manage invasive plants, pests, pathogens with compost. So that's going to be very exciting. All right. Well, ciao, everybody. Have a great weekend.
Ciao,
See
guys.
y'all.
Thank
Bye.
you.
See ya.
You